Saccharomyces cerevisiae is a species of yeast. It has been an elementary for winemaking, baking, and brewing for a very long time. It is originally isolated from grape skin, seen as a thin white film on the skin of some dark-colored fruits, among the waxes of the cuticle. Saccharomyces cerevisiae is one of the most studied eukaryotic microorganisms in molecular and cell biology. It is the cause of the most common types of fermentation.
Saccharomyces cerevisiae is described as a eukaryote microorganism and known as baker's yeast. This unicellular microbe is a yeast that belongs to the fungi kingdom. This globular-shaped microbe has a green-yellowish color. As a eukaryote microorganism, it has complete membrane-bound organelles. Majority of metabolism process occur in the mitochondria where there is no oxygen present. It is best cultured in neutral or slightly acidic pH environment in an aerobic condition with temperature of 28 degrees Celsius to 30 degrees Celsius, and most important, with adequate nutrient supply for it to grow.
In the environment, yeast cells are usually found ripe, dark-skinned fruits such as plums or grapes before the fruits mature, as they are almost free of yeast after maturation. Since Saccharomyces cerevisiae is not airborne, it requires a medium to move. Queens of social wasps living through the winter as adults can shelter yeast cells from fall to spring and transmit them to their offsprings.
Since the skin of that grape rich of sucrose, so as we have studied in simple and complex carbohydrate, the disaccharide sucrose contain combination of monosaccharide glucose and fructose. In the first step of alcoholic fermentation, in the presence of water and invertase, sucrose will be break down to fructose and glucose, so they produce 2 mole of glucose in this process. Each glucose molecule is broken down into 2 molecules of pyruvate, known as glycolysis process. This type of process is exothermic reactions. The energy from these exothermic reactions is used to bind the inorganic phosphate with ADP to produce 2 moles of ATP energy. Redox reaction also occur in this process, which is NAD+ donate their electron and becomes neutralized in the NADH form. So the last one is each pyruvate is converted to ethanol and carbon dioxide. This reaction occur in 2 steps. First step, pyruvate decarboxylase remove the carboxyl group to produce carbon dioxide. So the product from pyruvate is acetaldehyde and carbon dioxide. Second step is catalyzed by the alcohol dehydrogenase in order to convert acetaldehyde to ethanol. This process regenerate oxidized NAD+ and NADH remain in the cell. So, to conclude, the product is ethanol and carbon dioxide.
Saccharomyces cerevisiae start to break down the glucose. Glycolysis occurs to convert the glucose to form 2 pyruvate molecules. These 2 pyruvate molecules will undergo pyruvate decarboxylase, which is removing the carboxyl group to form carbon dioxide and acetaldehyde. Then acetaldehyde will gain hydrogen by oxidization of NADH and will produce ethanol. So this is how Saccharomyces cerevisiae metabolic pathway to produce alcohol.
Manipulation and microbial growth and activity. Factors that influence microbial growth. Nutrition. Mold are the least demanding nutritional-wise, followed by yeast and then bacteria. Second, pH. Yeast and mold have much greater tolerance to lower pH. Optimum pH for yeast is 4 to 6.5 pH. Next, water activity. Yeast bear environment of less water available than do bacteria. Next, available oxygen. Yeast will grow in the absence of oxygen, but more slowly than if the oxygen were present. The maximum temperature for yeast to growth is in range 37.5 to 39.8 degrees Celsius. The optimum temperature is 30 to 35 degrees Celsius.
Created using Powtoon.